Th Thank you, David. Um, I'll turn off my camera to save bandwidth, and I have control so I can proceed. So, hello and welcome to today's webinar on considerations of for bioavailability and bioequivalence studies of age-appropriate pediatric formulations. Today's presentation will have more a strategic focus with the aim of providing you with a suite of ideas that may be adapted to your specific program based on the risk and the pediatric clinical development options that may be available to you. Trying to advance, see the cursor moving. Okay. I'm still not able to move the. Okay. All right. Now it's working. Okay. So for today's discussion, the general outline for the webinar will focus on general pediatric clinical development options that are typically available to drug developers and also recommended by regulators. We will have a very short but high-level discussion of pediatric clinical pharmacology and the concepts behind selecting pediatric doses. These concepts are important foundation of pediatric drug development and sometimes are misunderstood, so it's important to clarify those. We'll focus on clinical bridging considerations for age-appropriate pediatric formulations, and I will try to clear some misconceptions about extrapolating bioavailability and bioequivalence studies, which are typically conducted in adults for pediatric formulations, and under what circumstances that extrapolation becomes problematic. We'll focus on that. And finally, we'll discuss what else we can do to ensure bioperformance of age-appropriate formulations by implementing certain assessments as part of the clinical program. It is important to note that today's webinar will focus on immediate release dosage forms. Modified or extended release dosage forms are a topic by themselves, so we'll not focus on those today. And I'd like to acknowledge Jack Cook. He's a colleague of mine at Pfizer who was very useful in providing some valuable insight to some of the concepts that are being presented today. So the design of a pediatric clinical development plan starts with the consideration of the pediatric study decision tree. So this tree was originally introduced by Dunn et al. Sometime in 2011 and was adopted by in a slightly different form by the FDA guidance that was issued for in 2014. So the decision tree poses two fundamental questions with respect to pediatric population or the target pediatric population. Is the disease progression similar in children relative to adults? And do children respond similarly as adults to the drug intervention? Depending on the answers, and followed by another question, which is whether the exposure response, which is represented by ER in this decision tree over here, is the exposure response similar to that in adults? So depending on the answer to these specific questions, you could end up considering three main options. Option A, which is akin to a full development program where you conduct PK studies followed by confirmatory safety and efficacy studies, which is very typical of what we do in the adult programs. And in the option A, you have answered no to pretty much all the questions that are asked of the program. So there's very little you can do to extrapolate. So you have to do pretty much the entire development program in pediatrics. Option B, where you conduct limited PK/PD studies using PD assessments, and then you follow that by additional PK studies if necessary, and safety studies for confirmation of safety. And option C, which is I would consider one of the most abbreviated pediatric clinical program, where you conduct PK trials to confirm exposures and then follow up with a safety study, because you can extrapolate a lot of the efficacy as well as extrapolate the exposures or systemic concentrations that you have observed in adults directly to pediatrics, and hence it allows you to do an abbreviated clinical program. So depending on where you land, you have multiple options. And on the right side, you have scenarios that I have put together, which again reflect these options A, B, and C. So if you can see the scenario 1 is where it follows the kind of an option A, where you do pediatric PK studies, you do dose selection, and then you follow it by pediatric safety studies. Scenario 2 is more similar to like option B, where you do PK/PD studies, you do select the dose, and then if necessary do additional PK studies, and again that's a caveat that if necessary, and then follow it up with pediatric safety studies. And then scenario 3, where you do just PK studies and follow it up with pediatric safety and efficacy studies. And this again is similar to option A, where you are actually extrapolating very little from the adult programs. And scenario 4 is kind of a hybrid version of all things being done together, where you are doing pediatric PK, safety, and efficacy study in one single trial, and then you are selecting the dose at the end. So, so these are kind of the scenarios, and there could be variations of these scenarios that you could encounter, but these are kind of the main pediatric clinical development strategies that you will probably adopt depending on which branch of the decision tree you land under. Now, one key thing you have to remember is, before we start even the PK studies, if we are conducting PK studies or start off any pediatric clinical study, you predict the dose using a suite of clinical pharmacology principles and tools which needs to be done before we start any pediatric trial. So this is kind of fundamental to the overall clinical development program, and the dose selection gets refined further as we collect PK data in the pediatric populations, and we generate the safety and efficacy data. A concept that we will be discussing in a lot of detail is the point of whether we use enabling formulation or the eventual age-appropriate commercial formulation as the part of the pediatric program. So you use enabling formulation when you don't have the commercial formulation available and you want to still start the pediatric PK study. So you use what we consider a fit-for-purpose formulation. And commercial formulation, goes without saying, is kind of the final formulation that you will commercialize for the pediatric population. So if you use enabling formulation, you are likely to require a clinical bridging BA/BE study or some form of in vitro assessments to bridge the enabling formulation to the eventual commercial formulation. So we will be discussing this in detail as well. One important note over here, that PK-only pediatric studies are hard to do as they offer no benefit to the participant. And it is very rare to conduct trials in healthy pediatric volunteers. So as much as I say that pediatric-only PK studies are hard to do, sometimes we are left with not many options. We have to do that. And when in the case where we do conduct PK-only pediatric studies, we have to offer options to the pediatric patients to enroll in long-term studies or allow them additional considerations if a PK-only pediatric study is done. Of course, healthy pediatric volunteers, studies in healthy pediatric volunteers are very rare, and I doubt it'll in the future we will be conducting any healthy volunteer studies in pediatric populations. All right. Okay. So as I mentioned, before we start pediatric clinical studies, we undertake a dose prediction, and it is conducted for pediatric populations using fundamental clinical pharmacology principles. So we'll briefly touch upon some of the main concepts of dose selection and what parameters are considered important for extrapolating PK in children. So as you are probably aware, pediatric patients have lower clearance relative to adults due to multiple factors such as organ size, maturation of clearance pathways, etc. And as pediatric patients grow, their clearance approaches the adult clearance. One point of clarification here: when I say lower clearance or when I say clearance, I'm referring to the total body clearance and not the clearance per kilograms that we sometimes encounter. For all practical purposes, when we are extrapolating and predicting doses in pediatric populations, and we are extrapolating clearance, we are extrapolating total body clearance rather than the per kg clearance that you may have seen. So all reference to clearance from this point onwards in this webinar is referring to the total body clearance. As I mentioned, as pediatric patients grow, their clearance approaches the adult clearance, and there already exists evidence that adolescents have clearance approximating adults. The PK parameters that are of importance and we typically extrapolate are clearance and volume, and these are extrapolated from adults using allometric principles to determine the dose. And we also use PBPK-based models which account for ontogenic differences, and they have been successful in applying. In summary, whether you use allometric approach or a much more comprehensive PBPK-based approach, these approaches have been very successful overall in predicting doses or extrapolating exposures for pediatric population. The PK studies conducted in the target pediatric population allow independent estimation of clearance and volume. And now a small technical note about the estimation of clearance and volume that we obtain from pediatric studies. That for orally administered drugs, clearance and volume represent apparent parameters, where clearance is clearance by F, where F represents bioavailability, and volume is volume by F, where F again is bioavailability. It's important to recognize that because for orally administered drugs, F cannot be distinguished from clearance and volume and is always included in the estimates of the apparent clearance or volume as the ratio. So it's important to recognize that as I continue discussing this concept over here. So as we collect PK data in the target pediatric population, whether we do dedicated PK studies or whether we do a sparse PK collection in the safety and efficacy trials, where we will analyze the PK using population PK analytical methods, what the estimates for the parameters, PK parameters that we get for clearance are the apparent clearance and apparent volume. The apparent clearance is the key parameter that determines overall systemic exposures as measured by the parameter of area under the curve at steady state, which is AUCSS, as is typically represented, and that relates to bioavailability and dose and clearance by the equation provided. So this is a fundamental equation in pharmacokinetics and is used conceptually in the extrapolation of PK from adults to pediatric population. So once the apparent clearance and volume and the eventual dose that targets that provides the target systemic exposures in terms of AUC has been identified for a target pediatric population based on PK studies, either dedicated ones or in safety and efficacy trials. Once we have determined that, all developmental aspects that may lead to differences in clearance, volume, and bioavailability in pediatrics relative to adults have actually been accounted for. So once the apparent clearance and volume has been determined in children along with bioavailability, because remember, bioavailability is part of the clearance and volume. We have accounted for the developmental differences between children and adults. And I repeat that because this is one of the most important concepts here, because from that point onwards, once you have those estimates, the ability of the pediatric dosage form to deliver the requisite drug dose to achieve the target exposure is of more significance than the pediatric maturational issues. So this is kind of the key message on this slide is that once you have determined the dose that achieves the target exposure, of course you have, and that is dictated by the apparent clearance and volume identified. You have accounted for the maturational issues that typically lead to the difference in exposures between adults and the target pediatric population. So this is going to be a recurring theme in this webinar, and we'll come back to this as we go along. So, as I mentioned, the need for clinical bridging for age-appropriate formulation is typically required when you use enabling formulations, and when you end up, and depending on what the formulation type is, what your drug properties are, you may end up requiring clinical BA/BE studies. I bring up an exception, which is highly soluble drugs formulated as solutions or as very rapidly dissolving solid dosage forms with inert excipients. I think the important qualifier here is inert excipients. May not require clinical bridging studies. And again, this is the topic we'll discuss this in shortly. But if you are doing enabling formulations, you have to do some form of bridging. Whether that is based on in vitro data or for clinical data will depend. If you use commercial or market image formulation for the entirety of the pediatric clinical program, and by that I mean whether irrespective of what scenario you follow over here, whether whichever clinical development pathway you choose, if you use your commercial formulation for pretty much the entire program, you will not require a clinical bridging bioavailability or bioequivalence study, because your exposures generated with that formulation during the conduct of the pediatric program would underwrite the formulation that will be eventually commercialized. So the use of the enabling formulation for the entirety of the pediatric clinical program, as I mentioned, will require clinical bridging BE with the commercial age-appropriate formulation. Switching from enabling formulation to the commercial age-appropriate formulation during the clinical program may require a BA or a BE study depending on the timing of the switch. So the important key qualifier over here is I'm distinguishing between a BA or a BE study when you are switching between between the enabling formulation and the commercial formulation during the conduct of the program. And I'll quickly refer to the definitions of BA and BE in the next slide. One thing I forgot to clarify, that when we are talking about the enabling formulation requiring a BA/BE study, if you use enabling formulations throughout your program, the eventual clinical study that will be required to bridge would probably be a bioequivalence trial. And the distinction that I'm drawing between BA and BE, we'll discuss in the next slide. So a BA study, as defined, by in the regulatory guidances, is where a formulation provides an estimate of the relative fraction of the orally absorbed dose that is absorbed into the systemic circulation. So a BA study, for all practical purposes, is an estimation of the relative bioavailability between the two dosage forms. And it usually is adequate to decide if a bioperformance of a new dosage form approximates that of the reference dosage form. So you are not applying a strict criteria of demonstrating equivalence. The BE study, on the other hand, is where you have a clear criteria. It's a comparative test. It's like a BA study, but it is a comparative test, and you're declaring equivalence based on the BE criteria, which is the 80 to 125 that we typically use. And this is necessary to conduct a BE trial when it is necessary to confirm that the new dosage form is equivalent to the reference dosage form. So there is a slight difference in when you use a BE study in context of the pediatric development program. Note that important to note here that the BA or BE studies assess the relative performance of the age-appropriate formulations in adult healthy participants, and we'll discuss more on this topic later, because BA/BE studies in healthy pediatric participants are very rare and unlikely. We will unlikely to ever conduct those. So you've heard about enabling versus commercial pediatric formulation for pediatric studies, and there is no universal approach. The choice of enabling versus commercial formulation depends on several factors, and here is an example of a biopharmaceutic risk assessment algorithm to determine which is better to initiate pediatric clinical trials with a clinical enabling or commercial formulation. The choice of that depends on, of course, there is a timeline and cost of development considerations, and there's also the risk that is associated with using an enabling formulation, which is dictated by the target population, the biopharmaceutical and PK/PD properties of the drug, as well as taste considerations. So this algorithm is not the only consideration, but this is an example of of things that you should consider, which to choose whether you want to use a commercial or an enabling formulation. This algorithm is specifically focuses on minimizing the risk of failing to demonstrate BE between enabling and commercial formulations. So as I mentioned earlier, no bridging is necessary if a commercial age-appropriate pediatric formulation is used for the entirety of the clinical program. And this is, again, irrespective of which scenario you have over here. So bridging with a pivotal BE study becomes necessary if enabling formulation is used for the entire clinical program. So you're using an enabling formulation at the end you want to market a commercial formulation which is different from the formulation used in the clinical program, then you require some form of a BE study. Now, you could have a situation where it's a highly soluble drug and you're not using any excipients, and you may be able to you may not be required to do a pivotal BE study, but that's a case-by-case basis, so we won't dwell on that too much. But if you have the enabling formulation and you do a pivotal bioequivalence trial, you still have to make sure that you don't need additional data if your mode of administration is different between the clinical formulation and the final commercial product. So that's another consideration you have to account for when you are designing the bridging strategy for the eventual commercial formulation. Now, the switching of formulations during the pediatric clinical program, and this happens very often, it happens more often than we actually predict or expect. There are many options possible, and I think this will require this will kind of take most of our time for the discussion. Because when we switch between formulations, the type of bridging that is required will depend on the timing of the switch, the formulation type that we are switching between, and the PK/PD and the biopharmaceutic properties of the drug. So the options that we typically have for bridging between formulations when we switch between the clinical during the clinical program, and we are doing the switch between clinical studies prior to the pivotal safety efficacy study, we have the option of using in vitro dissolution-based bridging, and this is definitely applicable to highly soluble drugs, which are formulated as solution or very rapidly dissolving formulation. And also, you have to remember that the formulation excipients do not alter with the physiological conditions affecting the drug absorption. So that's an important consideration when you're using in vitro dissolution-based bridging. PBPK-based modeling and bridging based on that is another option, and this is applicable to drugs where adequately qualified PBPK biopharmaceutic model exists. The approach requires substantial amount of prior data necessary to establish and qualify a PBPK model and will require prior planning. So if we have that, this is again a unique approach that can be used for bridging. And the relative bioavailability study with acceptance criteria specified based on the tolerance that we can have from deviating from the target exposure, considering the safety and efficacy. So an example criteria is a point estimate of the geometric mean ratio is contained within 80 to 125%. And if you have the new formulation that you are planning to introduce, if it meets that criteria, that's acceptable to be used in the subsequent study. So this is again another option that we have that we can use for bridging. So remember or recall that I'm using the term relative bioavailability rather than bioequivalence because and the reason for that is that we are still going to do additional studies with that new formulation. So a demonstration of an equivalence is not necessary at that point because the data in the pivotal trial will underwrite the exposure, the safety and efficacy of the exposure that is achieved with the new formulation. So the advantage of initiating the pediatric study with an enabling formulation, in spite of the fact that we know that we will require some form of bridging, is that with an enabling formulation or a fit-for-purpose formulations, it allows for a quicker start of the pediatric program. It minimizes upfront cost, and the development of commercial age-appropriate formulation can be staged and be informed by the emerging data such as taste and palatability. This also allows the assessment of the commercial or market image formulation in the target pediatric population. So note that bridging at this stage, as I mentioned earlier, only needs estimation approaches. The confirmation that the age-appropriate formulation achieves target exposures is established in the pivotal study using the sparse sampling and population PK analysis. The safety and efficacy profile in the target pediatric population is underwritten by the drug exposures achieved with the age-appropriate formulation in the pivotal study. Now, this is a very tricky switch of formulation approach where you do the switch in an ongoing study. I start off this slide by saying it should be avoided because I get nervous about this sometimes. However, sometimes it's unavoidable. And what I mean by an ongoing study is where actually the study is ongoing and you want to switch the formulation. Because the main reason why I get nervous about it is that if exposures are significantly different for whatever reason between the two formulations, it can confound the results from the pediatric safety efficacy study. There are exceptions, of course. If you have a solution form which is not using any excipients that can have an adverse impact on the absorption of the drug, it may not be that tricky, and it may be entirely possible to do that. But it depends. I think that the easy way to answer that is it depends. It depends on the risk profile of your drug, of your formulation, and of course the target population. If it is necessary to do that switch, which in an ongoing study, the bridging that we do should be confirmatory in nature. The bridging should consider a combination of the approaches, which is the bioform risk. Is your drug highly soluble versus poorly soluble? Is the formulation a solution versus a simple solution versus a taste mask solid dosage form? What does the in vitro dissolution-based data say in terms of similarity or equivalence? And it does require that the formulation excipients do not alter physiologic conditions affecting drug absorption. We can also use PBPK model if one is available, and under certain circumstances, it may be we may be required to do a confirmatory BE study before we actually implement that switch in an ongoing study. Once that switch is done, we can of course collect sparse PK before and after the switch, so that we can analyze the exposures for the two formulations in a population PK analysis. Extrapolation of the bridging BA study results to pediatrics, and this is a very dense topic, which sometimes people get very leads to a confusing set of understanding or conclusions. So I think this deserves a discussion of its own. So as I said, BAB studies for age-appropriate pediatric formulations almost always is conducted in adults. Conduct of such studies in pediatric populations without benefits is unethical. And the main objective of conducting BAB study in adults are to estimate the bioavailability and assess equivalence when applicable between the test and reference formulations. Detect formulation variables that may affect drug release from the immediate release dosage forms. Hence, the adult BAB studies of pediatric formulations are essential to rule out any bioperformance differences from a drug release perspective. If a difference is detected between pediatric formulations in an adult BAB study, then differences can persist in the pediatric population, or they may get even magnified in the pediatric population. If no difference is detected in adults, it is reasonable to assume or extrapolate the results to pediatric populations, provided physical properties and solubility of drug substances used in the age-appropriate formulations does not differ significantly from reference, and no excipients or formulation strategies are used that can interact differently with the pediatric gastrointestinal physiology relative to adults. So these are the two important caveats that we have to remember when we extrapolate, or we assume an extrapolation from the adult BABE trial to a pediatric population. The BABE studies of pediatric formulations conducted in adults demonstrate one thing and one thing only, that the drug release properties of the test formulation are similar to the reference formulation. And extrapolation necessary, and this is another important thing, extrapolation is necessary only if age-appropriate formulation is not evaluated at all in the pediatric population. So if after the bridging trial you do generate PK data in children, then you are not really, and you confirm that the exposures are as expected, then there is no extrapolation necessary. You just underwrote the introduction of the new formulation using the the bridging strategy into the pediatric population. Issues that can confound extrapolation from adults to pediatrics: drug substance differences. If you change the physical form, if a different salt is used, a different particle size is used, and these usually tend to affect the intrinsic solubility of the drug substances, and this can have an impact because of the physiological differences between adults and children. So this is something that we have to be aware of. Drug formulation, the dosage form release profile is different, or the mechanism of release is different, and we use excipients which can change GI transit time or interact with GI transit times, affecting release. So these can also affect or confound extrapolation from adults to pediatrics. So these are the two big ticket items I think we all should consider when we are considering extrapolation from adults to pediatrics. The last bullet on this one is what I'm calling confounded confounders, and usually these I club these under this category are age-related ADME effects, disease progression, physiologic factors, and so many other things that are typically get cited in the literature sometimes. The reason why I call them confounded confounders are, and I'm going to repeat what I said earlier, that once the apparent clearance volume and the dose that provides the target systemic exposures has been identified for a target pediatric population, all developmental aspects that may lead to differences in clearance volume and bioavailability in pediatrics related to adults are typically accounted for. Hence, if an age-appropriate formulation does not lead to a targeted exposure in a pediatric population, it is probably the formulation and not the target pediatric population. So we need this is the thing we need to recognize, and we need to focus on as we understand our risks of the formulation that we are introducing and the clinical studies that we are conducting. Hence, we have to focus on what property of the drug substance and drug formulation that we can interact differently with the pediatric physiology to confound the extrapolation from adult BA/BE studies. So this is something that if there's one take-home message, this is the most important take-home message that you can have from this webinar, is that how do we identify what is the risk that can contribute to a lack of extrapolation from the BA/BE bridging trials to pediatrics. The overall risk factors that actually can confound extrapolation of adult BA/BE studies is an evolving area, and more research is needed in this area. But as I mentioned in the previous slides, there are two main categories that you have to focus on. It's the drug properties and the formulation properties. And the additional assessments, if there are additional considerations that require additional investigation, then those additional assessments should be incorporated in the pediatric development program. In pediatric programs where a formulation is switched during the development, the PK data is collected in pediatrics for both formulations and can be used to estimate bioavailability using population PK approaches. And I'll quickly go over an approach that can be used. And of course, PBPK simulation-based approaches, and this is again an area of active research which I believe is very promising. The estimation of bioavailability may be adequate under certain circumstances, especially when you are testing both formulations in the target pediatric population, as the adequacy of the exposures for the safety and efficacy is underwritten by the acceptable safety and efficacy results from the trial. So remember, you are testing both formulations, you are estimating what the bioavailability between the formulations is, and eventually you will have the acceptability of the safety efficacy results from the trial as well. So there are multiple data points that you are generating that will allow you to tell whether a formulation is acceptable or not. And it's not a single bioequivalence trial that will do that for you. So this is the example I wanted to quickly talk about. This is using a population PK approach where you can generate relative bioavailability data in pediatric populations. And this can of course be applied as long as you have PK data for both those formulations in pediatric subjects. And there are some technical terms I will try to explain to the best of my abilities, these are pharmacometric approaches. So, but these are very powerful tools which will allow us to estimate bioavailability of two different formulations in the target population. So of course, when we collect the data in population PK setting, we collect sparse data for both formulations in the pediatric subjects. And what we do with the data is we hypothesize two different models. So we usually have an overarching model which is adequate to describe all the data. We can call it an adequate or a final pop PK model. But what we do with that model is we hypothesize that the alternate model to this final model or the kind of reference model, that the clearance is difference, which controls AUC, or we have an alternate model where we say that and we have an alternate model where we say the Ka is different, or rate of absorption is different, which affects Cmax. Now you can test the superiority of this alternate model 1 and alternate model 2 with the reference model over here using very standard pharmacometric principles. Objective function is a statistical test to say whether a model is better at describing the data over the other. So you can test the objective function of model 1 and model 2 versus the reference model and see whether it allows you to conclude whether either of the alternate models are superior to the reference model. And then you also look at parameter uncertainty using non-parametric bootstraps, which again is a pharmacometric approach or tool which allows you to assess what the precision of your estimation is. So the sensitivity of the alternate models to detect differences in rate of absorption should be tested using parametric bootstrap, and that is what I mean by precision. These an important note that these assessments are best done a priori, that is before you actually do the trial, so that you know that when you have the data, you will be able to test the relative bioavailability of the two formulations. But in certain cases, if you have not done that, you can also do this post hoc to qualify whether if your conclusion is that the formulations are similar, that you had the sensitivity to detect that there is a certain magnitude of difference. So as I mentioned, the sensitivity of the alternate models to detect the differences is tested using parametric bootstrap, which is a method of checking the precision. And what is done is that you use the models assuming a certain difference, and where you assume a certain difference and you do simulations, and with the simulations you fit them using the reference model and the alternate models which I showed in the previous slide, and identify how many times you were able to detect the true difference, because remember you are simulating with a difference. How many times that you were able to detect the difference and the alternate model was truly superior. So this is basically a sophisticated power calculation where at the end of this analysis you can say that what is the likelihood that if your formulation was truly different by a certain magnitude, whether we can pick up the difference. So for this example that I have over here, we have a power to detect a difference of beyond 80 to 125 of at least 70%. So this analysis is kind of important, especially when you are testing, when you are having a confounded situation where you are worried about extrapolating from adults to pediatric populations because of many of the reasons that we discussed earlier, and you want some more confirmation that the formulations are actually able to perform as expected in the pediatric population. So this analysis is kind of important to validate the results of your POP PK for a relative bioavailability assessment. So this is my final slide. So there is actually there's no one strategy that fits all. But what I mean by that is it all depends on what is the risk that we are anticipating, with whether it is with respect to the API, whether it's respect to the BCS class of the drug, whether it's respect to requirement of taste masking, solubility, excipients, formulation. We have to make that assessment at the start, and we have to develop our pediatric clinical development plan appropriately so that the risk is balanced out in the assessments that we target in our pediatric clinical development plan. So the pediatric development plan should integrate the development of the age-appropriate formulation in the strategy from the start, and the formulation switches during development should be carefully planned along with appropriate bridging studies. I'm using the term BABE studies more for bridging studies or bridging assessments, because sometimes you end up not needing a BABE study. And switches in formulation during ongoing pediatric studies require careful consideration of risk factors, and if necessary, a confirmatory BE study may have to be conducted prior to the switch. And extrapolation of BABE studies in adults should account for the typical risk factors and plan for confirmation in the ongoing pediatric studies using quantitative approaches. So with that, I'll end the webinar and initiate the discussion and question and answer phase.
Thank you very much, Vivek. And I have some questions coming up from the chat, and I think I'll start with the first question. You have shared in the slide deck a couple of times about taste. And from your experience, when and how do you collect the taste data?
So the taste data is one of the important considerations when you're designing the age-appropriate formulation. And again, to get a sense of taste, at least at Pfizer, we do the taste assessment early in the program, of course, and the taste assessment is done in adults initially to assess what kind of what is the taste profile of the drug, and then if taste masking is essential, so that when we going into a pediatric study, whether we end up using an enabling formulation or the eventual commercial formulation, at the start of the pediatric trial, we at least have some taste, assessments available, so that the enabling formulation or the commercial formulation has been designed so that it will have an acceptable profile in pediatrics. And we also collect pediatric data, taste assessment data, as part of the safety efficacy trials as well as PK trials if we conduct PK trials. So taste assessment is an ongoing process, but we do start in adults before we actually initiate, or we should consider starting in adults before we initiate pediatric trials.
Thank you very much. I think the next question is, do you consider different age groups when assessing product performance? And if so, how?
Yes, we do. That is again that is covered by the target population, and in terms of dose selection, the dose selection approaches that I kind of highlighted, those are applicable to all pediatric age group. They go down as much as low down to the neonates. Then for the neonates, it's a little bit more complicated, and we do end up relying on more physiological based pharmacokinetic approaches to predict doses. But the dose selection is applicable to all the way down to the youngest age group possible. When it comes to BABE strategies, they are also applicable to pretty much all age groups. With neonates, on the other hand, especially for oral formulations, there may be some additional considerations which I may have not discussed at today's webinar. But there are some additional considerations that you may have to account for in the youngest age population, but down to the typical age group of 2 or 3 months of age, the strategies that were outlined in the webinar are definitely applicable.
Thank you. And the next question is about the dose for pediatric studies. So oftentimes we estimate the dose based on adult data or from adult bioavailability study. And you did mention relative bioavailability study for comparing different formulation testing in peds. The question is, how about relative BE to compare the adult versus the pediatric formulation? When is it needed, and can it be replaced by PBPK modeling or in vitro study as well?
So when you say bioequivalence, the bioequivalence term, as I mentioned, it has a very specific meaning in terms of what from a study conduct perspective. But typically, when we are comparing adults and a pediatric population, in reality, we are actually looking at the bioavailability differences between the formulations, not formulations. I'm sorry. We are looking at the bioavailability differences in terms of the target exposure. So, majority of the times, we are targeting the same exposure in pediatric populations as adults, because the exposure is what dictates the safety and efficacy, and safety and efficacy once established in adults, if it can be extrapolated to children, as long as children are similar. Remember the clinical strategy diagram that we discussed earlier. If they are similar, then the exposure is similar, and from a BE perspective, you're targeting the same exposure. So, in a sense, the pediatric program in its entirety is basically assessing whether you have the same exposure as adults and whether that translates into the same safety and efficacy profile in adults. Now, if I understand the question correctly, that's how I would explain it and approach it. Now, do we need to conduct bioequivalence trials between a pediatric formulation and an adult formulation all the time? I don't think it is necessary, and it depends. And the reason I say it depends is that if you want to use the formulation across all age groups, the pediatric formulation across all age groups, you may be required sometimes to conduct a bioequivalence trial. However, I would like to make the case that that for a formulation, adult or pediatric, if you have established safety and efficacy in adults independently and safety and efficacy in pediatrics independently, and as long as they are approaching or having a similar range of exposures, which is within the range of acceptable, efficacious and safe exposures, I think you have enough data to say that the formulations is acceptable across all age groups as long as you meet those criteria. So, long answer, but there are a lot of ways to address that answer. But typically we don't run bioequivalence trials between adults and adolescents, adults and pediatrics. Sorry.
Yeah, thank you, Vivek. One more question on this. This regards to bridging studies. So, assuming that the PK pediatric study data is available for the proposed commercial formulation, have you seen instances where bridging study is required for 2 oral suspension formulations, one for single dose versus multi-dose, when we conduct the switching during clinical studies without BA or BE data?
So I didn't capture the first part. So what I understood was that you have pediatric PK data, and you have 2 formulations, 2 suspension formulations, and you want to switch between the formulations. Can you do that without a bridging BA/BE study?
Correct.
Is that correct? Okay. you do require, so if you want to switch, at the end of the day, it's a risk that you take. So if you don't do a bridging trial, means you don't do anything other than just introduce the formulation. That's a risk. At minimum, you have to conduct at least the in vitro dissolution work and make sure that you are generating enough information about the comparability of the two formulations. Now, granted, we are going from suspension to suspension. Depending on the excipients, you would not expect major differences between the formulation. So I made the case earlier that if you are going from solution to solution and the excipients there are no excipients that are impacting the physiology of the GI tract to affect absorption, yeah, you could definitely do that switch. I think the same argument would apply to a suspension, because it depends on whether you meet those criteria. But at minimum, you should do at least a dissolution assessment to establish a bridge, because the in vitro assessment is still a bridge.
Yep. Sounds good. Thank you. I think in the interest of time, I think we will stop accepting questions for now. If you have additional questions, please feel free to share with Mahalia. And again, I'd like to thank Vivek for your time and this excellent presentation. And thank you, everyone.